Okay, so today we are going to talk about the general structure of the lab report, which is almost the same for articles, for thesis, and the lab reports themselves. Okay, so here I have a nice article from the internet. It's a random article and it was published in Nucleic Acids Research in 2019. And we're going to dissect it so that you understand how everything is going on. Okay, so first of all, of course, you see the title. Then on the first page, you see the abstract, then introduction. Then they switch to materials and methods. Then they go to results section. And after they presented all the results, they switch to the discussion. So in this particular article, they don't show the conclusion part, but you could also squeeze it when you do the articles or thesis. You could squeeze it into your discussion section, and it might go as the small summary. But for your lab report, of course, we ask you to have it as the section, so you will need to write something like conclusion section in your lab report. Finally, we have the reference section, and here you go with all of your used articles or other sources. Okay, so now let's talk about everything in details. So here you see the title of this article, and it clearly states that it is about the long non-coding RNA MEC3, which is protecting the endothelial functions. And how it does it? It does it by regulating the DNA damage response. So it is a very active title for the article. And how do you understand it? So you understand it by the verbs used. So here you see the words protects and regulating. So what else you could write here to make it more passive? You could write something like long non-coding RNA MEC3 in the DNA response as the protection for endothelial function. Okay, so this is a very passive, and it actually does not tell you much about the article itself. When you use the active form of the title, it gives you much more information, the kind of vector of your paper, so that the ones who read it in the title section's website, they will be able to identify the articles that are most interesting for them. So the next part, so first of all, of course, here you see the active title. Second, you see the abstract. And in this case, for this particular lab report of yours, we do not ask you to write the abstract, but in general, you need to write it. So what is abstract? Abstract is a short version of your article. So in this abstract, you should be able to squeeze all results and all discussion that you have together with your conclusion part. Why you would need it? Because person who just took the articles and starts reading it, they should be able to identify if this is of their interest. If it is not, they will need to understand it from this abstract. Why? Because all of us have very limited time for reading, and of course you don't want to spend a whole hour for reading an article that is not in the field of your interest. This is very important part, but of course for the lab report for MB2 you don't need to write the abstract, but in general you should be able to concisely explain your article. Okay, so now let's talk about the introduction, because this is something that you should be familiar with, that's first, and second, you should be able to present some information, wide background information for the reader, so that they don't get clumped with all the possible information that is related to this field. They should take a very concise bite of the information from you. And this information is coming in your introduction section. As always, you start with broad information, then you switch to a much more focused one, and in the last paragraph you should tell about your problem of the article, the hypothesis and the aims. So for example, in this particular article, they say that the Mach3 is actually something that is related to many cellular functions, but they state the problem that it remains unknown how the Mach3 determines the transcriptional output. In this case, problem does not mean that they have a real problem that they cannot solve. No, this is the question why they approached this problem with their hypothesis. In the introduction, you should be able to give hypothesis plus aims of your project or of your paper. Okay, next you switch to the materials and methods, and in this section you should start with the reagents used, then all possible methods or techniques that you used to approach the results that you obtained in the end of this project. And as you can see, it's a very extensive explanation of the methods used. So for example, RNA immunoprecipitation, the real-time qPCR, cell cycle analysis, Caspase, activity, assay, etc. etc. So they have a very wide range of all these methods and techniques used, and they explain it so that everyone, everywhere in the world should be able to understand how it worked, why they obtained the results, and also they should be able to repeat it. Why it's so important for other scientists to repeat your experiments and the whole project. So repeatability is the main criterion for a good science. Okay, so if you read an article that cannot be repeated, it means that this article does not worth anything. This is a very important part. That's why many scientists, they pay huge attention to materials and methods. All we know from the lab books that you've written during the courses is that it's something that you usually don't spend much time on. But when you do the science, the real science, you should be writing something that will guide others so that your experiments are repeatable. Okay, so next we switch to results, and as in materials and methods, they have the subsections. So what are these subsections? These are the sequence or the series of experiments that were done during this project. Of course, the project is not the single experiment, it's a really consequent flow of the experiments. And as you see, they present all the results in this section. So they have subheadings, and you probably need to have subheadings too, because you had week 1, week 2 and week 3. For every week you had different aims, or let's say like this, for different weeks you had different results that were showing the methods that you used for approaching your aims. So 3 different aims, 3 different results, and 3 different discussions for these 3 weeks. Okay, so also what you see here, you see some links to the figures. So for example, you see figure 1a. So of course figure 1 is the number of the figure that you're going to use, and A is the panel of this figure. So here you see the figure 1, and here is the panel A. So they also have panel B, D, C and E. Now let's take a quick look at the figure and how it's explained. So we have a legend here. Well, it's not legend, it's actually a small explanation which is the figure caption. Okay, so you should always start with the name of this figure and a very short explanation of what is shown on this figure. And as you can see here, it is the expression of Mach3 that is induced by DNA damage in a p53 dependent manner. So by reading this only, you should be able to understand, okay, so here I should see something related to the p53 activity and the related expression of Mach3. Then they give quick explanation for each panel. And what is needed for this part, for the figure caption, you should give as much information as needed for anyone to understand it if they just see it without the article. Let's say arbitrary person sees these panels and they read it, they should understand, okay, so here we see the relatedness of the MEC3, so we have the increase of its concentration in the cell while we are increasing the doxorubicin. So doxorubicin, based on this explanation, is the drug that increases the DNA damage. And because we increase the DNA damage, the p53 dependent processes start and they activate the MEC3 expression. That's what you should understand from the figure and its caption. The same goes for all other figures, and let's see what they have. So for example, here on the panel A, you see the images of DNA tail lengths, which are related to the treatment with the TNF-alpha treatment. Here you see the volcano plot, and this volcano plot is the graph. You could write it as a graph or as a figure. Also you see some histograms and the comparison between different treatments. Again, you have the subdivisions or subheadings for different experiments. Okay, so here is how the results are written, and of course when you write the results, only the ones that you obtained from your experiments should be written here. All your thoughts, all your hypothesis related stuff should be given in discussion. Everything that is related to your experiment and the output of the experiment should be in the results section. Okay, so when you finished with the results, you proceed to the discussion. Okay, so in discussion, you actually claim something and you try to prove it. Okay, so the discussion is the main part of your article, lab report, or the thesis. So this discussion is something that proves your hypothesis. It does not mean that if the results disprove it, you should still prove the hypothesis. If your results say that your hypothesis did not work, in the discussion you give the disproofs for your hypothesis. Let's say you have two different situations. In one, your results were proving the hypothesis, so you write something like, according to these results, we obtained the information that gave us the basis for proving the hypothesis, and vice versa. Although we hypothesized this, and this, the results said that we were wrong. But because we are flexible and we know that these processes does not happen in the cell, we try to re-hypothesize this and this and this. And due to this new hypothesis, the results might be explained in this and this and this. And for proving this hypothesis, we need to do other types of experiments and obtain such and such and such results. So this is, the discussion is when you explain why you obtained the results and how they are responding to your hypothesis. Again, as I said before, the summary might be the last paragraph of your discussion, or it might be a subsection or section, which is the conclusion. And for example, in this article, in the summary or in the conclusion, they say that they demonstrated, they found that their data highlighted something, and because they had this and this and this data, it suggested this and this and this. So this is, the conclusion section is only about what you achieved, what you get, and how you explained it. Okay, so In general, it just means that you achieved the goals that you set for yourself in the beginning of the journey. Not the journey, but the project. Okay, so you finish with the references. And the style that we ask you to do is the Harvard referencing system. Okay, so what is Harvard referencing system is the start with your authors, then you have the year of the publication, then you have the name of the publication, the journal, the issue of the journal, and the pages of your article in this journal. And this is the general rule for Harvard referencing system. If you are referencing the sites, they have a very specific way of declaring it, but you should be referencing everything that you used in your lab report. So this reference actually gives you kind of the key to the projects that were done before you, and based on these projects that were done and have proven their claims or their hypothesis is the basis of your project. So you cannot write an article without the references. You cannot write the lab report without the references. Why? Because this is the consequent flow of the projects. So one project is related to others, and this relatedness should be shown in your references. If you have a very huge data that you need to show, but you are limited in pages, you might put some of your results in the supplementary data, but of course for your lab report you won't have this session. You also will not have the acknowledgements. In usual acknowledgement just show what units, what people were involved in the project. Sometimes you write something about funding, about the conflict of interests, etc., etc. So you should be stating this everything because you want your work to be trusted. So this is the end, and if you have any questions you could always email me and ask for hints or tips.